Hello, everyone. My name is Barbara Olack, and I work at City of Hope with the Integrated Islet Distribution Program, which is an NIH-sponsored program that allows human islets to be distributed to investigators across the country. This is going to be a very, very informal workshop. And the first thing I'm going to do is I'm looking around at the room and I see a number of people that do not need to be here. Okay, so this is basically for people that are not used to working with human islets, have worked with maybe rodent islets in the past and are trying to get into working with human islets, or new investigators that have never worked in islet research before. You're very welcome to stay, but I will not be offended at all if you get up and leave in the middle of the talk, okay? Because this really isn't going to be a talk. It's going to be more like a give and take, question and answer period, just more like a workshop than a presentation. So welcome to everyone. There is a list, a sign-up sheet that's going around. If you haven't signed it yet, we'd really appreciate it. On there you can put your email address in case you're interested in getting any more information about receiving human islets for your programs. We will contact you and give you some information that you may not receive here in this talk, because this is not really about our program as much as just using human islets and working with them. There's somebody else that can leave. Just a little background about myself. I've been working with human islets for many, 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 many years, since about 1977, and started at Washington University with Dr. Paul Lacey, Dr. David Sharp, and it was in some of the first islet transplant programs in the country, or in the world. So anyhow, please find a seat, and like I said, we have some brochures about our program sitting up at the end of this session. If anybody's interested, we do have some counting books, some folders, pamphlets that we've put together that will you can actually use as a training manual in your laboratories for learning to count and grade human islets. And we didn't really think there'd be this many people here today, so we have about 30 books, but we'd be happy to send you some in the future if you write down on the sign-in sheet that you're interested. So let's get started. Is that my little thing? I see. So the first thing I'm going to give you is a little bit of background information on isolated human islets. I'm going to talk about the process of getting those islets, about the importance of donors, how an isolation is considered successful, at least in transplantation, and the difference between human and rodent islets. And that's the first thing I'm going to ask. People that are here, like, why are you here? Are you using rodent islets now for research and you want to go to human islets? Are you a new investigator just getting in the field? So how many people have used human islets in the past already that have done this? Okay, so a lot of people. That's great. So you can probably leave. No, I'm kidding. You're welcome to stay, but don't feel bad if this is really boring to you. So I guess a lot of people, and are there people here that have been using rodent or other animal islets and would like to get into human islets? Anybody here doing that? Okay. Well, hopefully this will be something that you will be helpful for you, and maybe we can help you out as a program as well. So the process, the islet isolation process, I don't know how many people, I know there are people in this audience that have seen human isolations before, but it starts out with a pancreas retrieval, a cleaning and decontamination. Then you cannulate the pancreas. Enzyme is added. That's done with a pressurized pump that blows up the pancreas through the ductal system very slowly. After a while of digestion, adding the enzyme to the pancreas, it's cut in pieces. It's loaded into some kind of a chamber, and this is the Ricordi chamber that many of you are probably have seen in the past and used pretty widely around the world. After that, it's collected. The digest is collected at a certain point. It's collected in pellets, and you get a prep like this that is, the islets are stained in red, but as you can see, the majority of tissue that you receive is other pancreas tissue, acinar tissue, and other connective tissues. So we're interested right here in these islets. After this point, the purification process begins. And with this, for human isolations, most people now use a COPE, which is it's actually a blood purifying machine that was adopted was adopted by the islet community and adapted to human islets. And what it is a centrifuge that allows tissue and fluid to go in and come out at different points with a gradient. So there's a gradient that's added right here, and this is a Ficol gradient that's used in, has been used for years, and is also used in animal isolations to purify by density for the islets. Those are then spun for a while in this machine. The different layers are then collected at different volumes so that you will separate out the purified islets from the acinar tissue is the point. And this right here is the tissue that's left over at the end, and hopefully this will be, the majority of that tissue is going to be acinar tissue. So here's a graphic of what happens. The tissue goes in, the density gradients go in, it's spun. It spins for about 8 to 10 minutes depending on your protocol, and then it's released and it comes out as it lines up. The islets will come out at different gradients, or the tissue will come out at different gradients, islets usually being close to the top. This right here is a impure prep, and then hopefully after the purification process you'll get purified islets. Everything stained in red is our islets. So some of the factors that affect an islet isolation start with the donor, and these are some of the categories: age, body mass index, some medical history. Also, there is a big interest now in research for type 2 donors, which we as a program are trying to provide not only the islets but also the important information that goes along with that from the donor to ensure that it's really a type 2 donor and not just someone that has a high BMI, or whether it's a medically treated donor, whether they're on medications, things like that. Hospitalization medications affect the isolation as well as cause of death and the cold ischemia time. So if you talk about success of an islet isolation, that can change depending on what you're going to use the islets for. But based on clinical application, quantity is very important. Usually a center will get over 500,000 islets from an isolation to be used for a transplant. Purity, obviously for research, purity is usually very important. For transplantation, it may not be as important. Depending on how they're doing the transplant, if they're doing them encapsulated, then it again comes to a purity is very important. But for just a normal transplant, especially an autologous transplant for someone with pancreatitis where the islets are isolated, that in itself is not important at all. It can be as low as 50%. Viability, of course, is important like any kind of cellular work. And as far as potency, meaning not just the viability of the islets, but how they respond to their glucose, challenges. Unfortunately, at this point, I still don't think after 30-some years that anybody can totally agree on the best method for measuring potency in islet isolations or in islets, both human and rodent, I think. So right here, I just wanted to point out the differences between both human and rodent islets and the different things that they're used for. And this is mainly for the people that are working with rodent islets now and would like to go to human. Basically, their human islets are much smaller overall than rodent islets, and the average sizes are totally different. Your range of islet sizes in humans seems to be a lot larger than those of rodent islets. Of course, the donors are going to be heterogeneous for humans. Every donor's got a different background, a different age, a different weight, where if you're working with rodents, you can pretty much pick what you want. You can have the same breed, the same age, the same sex, everything's the same. So it makes it a little more difficult. The cellular architecture of human islets is different than that of rodent islets, where in rodents the beta cells are in the core of the islets, and in human islets they can kind of be scattered all throughout the islets among the other cells. The cellular composition is different. There's less beta cells. It's been shown in a number of different papers that there's less beta cells in an average human islet than there are in rodent islets, which then again influences the insulin content and the insulin production as well in human versus the rodent islets. So as you can see, as standard, the insulin secretion for human islets is lower with an SI of 6.3 compared to most rodents, which can be anywhere from 12 to actually can be a lot higher than 12. I've seen 30 stimulation indexes of higher than 30 for rodents. And there is, of course, differences with every human, but the averages, there's a lot less insulin per human islet. And you need to keep that in mind if you're in a program looking for human islets, that what you're requesting is going to measure up to what you've been using for rodent islets, because you're going to need more human islets overall to achieve the same kind of research results as you do with your rodent islets. Now this is down to more about the workshop. That was kind of the background. So this is some ideas and some suggestions. And this is based not just on my recent past, but also on the centers that we have worked with in the IIDP and the ICR, which was the Islet Cell Resource Center. And it's based on the different programs, from 6 to 10 programs at different times that we've worked with, of centers that do islet transplantation, for human islet transplantation, and then provide the excess islets for researchers. So one thing we're going to talk about first is proper sampling. And to me, I think that's the most important thing to success when you're doing any kind of a research project, because there are so many variables that can influence the number of islets that you're actually pulling out. And if you're trying to do comparable research, you definitely want to get as close to the same as you can. So first, there's a mixing technique. It's what sampler you use, and then your sampling technique. So first of all, for a mixing technique, people use different techniques, and like I said, this is based on the programs that we've worked with over the last 10 years to get a sample from an islet preparation, either one that you've isolated yourself or that you've received from another center or through a distribution program, to be able to get the islets that you need for your studies. So overall, inverting, if you have it in a test tube, either this size or a larger size test tube, inverting it back and forth or pouring it back and forth are two ways to make sure that you get a nice homogeneous mixture of the islets, of the different sizes and the different amounts. Now, also, we also like to, and I know this isn't always possible if you're doing your experiments alone, but to have a second technician help you take the sample is always important. I just found out, I think a week ago, that one of the transplant facilities that have been doing this for years somehow had gotten to the point where they thought they could just do it on their own. One person taking the sample by themselves, and found out that their samples were 1,000 times off, because by the time they got their sampler out, whatever that is, took their sample, even though they had mixed, they took their sample, all the larger islets had settled to the bottom of their flask. They were taking samples from the middle and only ended up taking a section of islets that weren't representative of the whole thing. So I know this sounds kind of a pain, but it's something that should work. And the biggest thing is don't swirl your preps, okay? Because the islets will tend to centrifuge, like we showed before for separation. The larger islets will go to the outside of whatever you're swirling. The smaller islets will tend to stay in the middle. If you take your sample from the middle, you're only going to see smaller islets. The next thing, is the samplers. And there's a number of different kinds of samplers. You can use a wide mouth syringe with a pipette aid type thing. Make sure if you're using human islets, I would suggest that you have filters on to help preserve your sterility of that. I know for a lot of transplant programs, they use these Drummond microcapillary pipettes, which are nice because it's just like a syringe. You push it, you pull it up, and it's set for the exact amount. It's a nice wide bore so that the bigger islets aren't going to get sheared when they pull through, and it'll pick up smaller islets as well. Oh, and for people that are writing notes, we are going to have a handout at the end of this that you're willing to you can take if you'd like to, and it will have different products where you can buy these different products. Not that we're endorsing anyone at all, but it's just different suppliers and different catalog numbers to help you out. Plus, there will be the book that I spoke about earlier. So don't go crazy trying to get numbers down. The other thing that you can use is a regular pipetter, and all I can say about that, those work fine, but make sure and get the wide bores. You do not want to shear off any islets, so make sure that your pipette openings are large enough. Not the little tiny Eppendorf pipette openers will not work for any islets, especially human, because they're a lot more fragile. So a sampling technique that many people recommend is that you take from your volume, you put it into and put the entire thing into the counting dish. You count the entire sample of what you've picked, and make sure that when you're counting, you count a representative number. So in most cases, counting 50 to 100 islets, if you're talking about a volume of, or a quantity of 100,000 islets, that's going to be what you want to do. Of course, if you're picking out and you're only trying to count 5 islets for, to put in a well or into some kind of an assay, you don't need to count 50 to 100 islets. But just make sure that your sample matches what you're trying to do with your research study. So a couple other tools that are used normally in human isolation and probably most rodent isolations and studies as well is an inverted microscope with a gridded eyepiece, something that gives you an orientation, and then a staining to identify the islets. So one of the first ways before dithazone was actually used as a stain, and I imagine most people use something like dithazone to stain your islets, is Ed Fink, who happened to work with Dr. Paul Lacey at Washington University, years ago found out by putting a green filter on the microscope that the islets would actually autofluoresce. So they would look like this brilliant gold fluorescing group of cells compared to the acinar that were really dull. And that was used for many years before the dithesone came. And it is something that if you are trying to use the islets that you're looking at in the experiments, it's something that you can work with. That way you don't have to add a stain that could change the viability of your islets while you're working with them. So it's one thing to keep in mind, meaning that old stuff isn't always bad stuff, new isn't always the best. The eyepiece, there's different kinds of eyepieces that people have. There can be these rulers. Well, that wasn't the right one. There can be these rulers. There can be just a round grid, or there can be a 10 by 10 grid. There's a lot of different things, and they all work. It's not a big deal. What you're trying to do is measure the size of your islets, because one difference in human islets, I think, compared to rodent islets, is that most people talk about rodent islets as 1 eyelet, 2 eyelets, 3 eyelets. With human islets, it's been called eyelet equivalents for about 25 years now. I think the first time that eyelet equivalents were determined and talked about at a meeting was at the first IPITA meeting, I think now 25 years ago. If it wasn't at IPITA, it was at the cell transplant meeting, but it was one of those two that I think the idea of an eyelet, a human eyelet isn't an eyelet. There was totally differences in the size of human islets, and that had to be somehow, for transplantation, that had to somehow be quantified. So what was decided on and pretty well accepted is a grid size, and this is an old, well, I'll point that out a little bit later. But right here is the grid that actually helps for orientation. The ones before were for sizing. This one right now is for orientation. So one thing that works very well is you can buy these polystyrene culture dishes to do your counting in. And it has a grid underneath it so that you don't lose track of what you're counting. And that's all it's for. Or you can get an etched mylar sheet and put it underneath whatever you count with. And then you move that whole sheet, and it will not let you lose your place. I know that people have stages on their microscopes that hold culture dishes or petri dishes where you can move that as well, and as long as you have some kind of a grid that keeps you in line, then that's fine. That will work perfectly fine as well. The dithesone staining is the typical stain that's used in most eyelet isolations and in most eyelet isolation centers. This is a standard recipe for this. I think it's going to be in the book that we will give you, but there may be other ones, but and there's variations of this. I'm sure every lab has their own little nuance that they use, but this works just fine, and it will stain the eyelets a bright red. And you can see here in these pictures that here's a very contaminated, unpure prep, contaminated with tissue that is not eyelet tissue. So most of this is acinar, the white, and it can be different degrees, either pre or post purification. And of course, this is a very, very pure prep of really nice human eyelets. So I'm going to just go over the tips for sampling again. This is a kind of cluttered slide, so I'm going to go through it really fast. Just try to use 2 people if you can. First person does the actual mechanical mixing, the second one takes the sample. It's better to invert or pour back and forth between containers instead of swirling. Use a wide bore whatever to take those samples. You don't want to fray the eyelets while pulling them through. It's better to pick up the exact amount of media and eyelets that you're going to count, because if you ever watch your pipette when you're dispensing eyelets, they tend to swirl up the pipette, and when you release fluid, but you don't release all the islets. The islets tend to go up. And if you do that, you're going to be counting a lot more islets than you actually have, because your islets per volume of media is going to be greatly changed to what you originally took. So that's why we recommend using one of the pipettes where you just draw up to your mark and you stop. If you use a regular pipette, and you draw up and then you have to let some out, you're not going to get a proper sample. So it's kind of a technique, and that's why the little Drummond thing that you just push and open up actually works better than trying to watch a pipette as you pull your volume up. And then, like I said here, it says here that the swirling can cause a problem with your actual sample. Some additional tips is to take the samples from the middle of your prep, not from the bottom or the top. Make sure that you don't have clumps in your prep when you're taking them. And this is for people that are trying to verify what they got from an islet distribution center, or if you're just taking your samples for your different assays or for your different experiments. Just islets tend to clump. Some are worse than others, especially human islets. If there's overdigestion in the process, you're going to have a lot more clumping. Sometimes it's just the nature of the donor. So make sure that you have a well-suspended prep before you take your samples. A single dilution is the best. So you're going to have to dilute your sample, but you don't want to dilute from one volume to another volume to another volume. Of course, the more you dilute, the more a chance for error. Then also we have some details on our website, which is right here, and there will be details in the books or the handouts that we have at the end of the talk. So this is the grid I was talking about that was done about 25 years ago at the IPITA. This has been modified a little bit and made a little more stringent, but this is basically the list of what's used. And some of these the factors, some of the ranges and the factors have been changed, but it's normally the same. This is how you figure out an islet equivalent. You do counts in different groups of islets. So you'll count your islets that are 50 to 100 micron, 101 to 150, and going all the way up. And like I said, some labs make this even more smaller increments. And then, oops, excuse me, I'm sorry. So let's see, how do I go back on this? There we go. And then there is a multiplication factor that is based on the volume of an islet as a sphere. Now, some people are starting to figure out, especially with electronics and computer imaging, they're being able to look at different sizes and not look at an islet as a sphere, because we found out now that, first of all, islets aren't spheres. They're not exactly a round or a spherical shape. Plus, they tend to flatten out when you count them in a dish. So you may be seeing something a little bigger. So it's just that this is something that's been actually accepted by the islet community, and until somebody comes up with a great new technology that will count it better, I think that most people are using this. And if I'm wrong, if there's something new out there, please feel free to let me know. I know people are working on a lot of things, and I'll show you that in just a little bit. But as far as overall acceptance, I think that this is pretty well. There's somebody else that doesn't need to be here. So what I'm going to do is practice makes perfect. And so here we have a grid of an islet, and this is actually comes from our book that we're going to give you. A grid of an islet isolation with different islets on it. This was actually used in a study long time ago to make sure that all transplant centers at the time in North America were doing things the same. And everyone had to do these counts and make sure that they came out the same in order to be part of a transplant program or transplant study. So if you look at this, each one of these little squares is a 50 by 50 micron size. Here are your islets up here. And what we're asking is number 19. If it's 50 by 50, what size would that islet be? Anybody want to guess? Yeah, that's right. So it's going to be, because it's less than 4 across and 4 up, but it's more than 3 across and 3 down. So the average size, and that's why we have these increments. Of course, you're not going to get the exact, you're not going to sit there and count the exact size of the islet if you're hand counting, but it gives you an idea. This next one is for number 2, right here. And this one is pretty small, but it's bigger than 1 square by 1 square, so it's going to be between 50 and 100. It's very simple. It's just math. You count each of these on like a hematocrit. I don't even know what those are called. Where the little counters that you use.
Hemocytometer.
Thank you very much. Well, the hemocytometer is the grid, but the counter is for counting different blood cell types. If you use those, then that way you can have your different ranges, and you just have to get good with your fingers and practice a little bit. Now we get into the automated technology that's coming up, and there's a couple different things here that I just want to point out. I'm not going to go into detail because this is mainly used by islet transplant centers that isolate huge amounts of human islets at a time for transplant. But this is done by using little microspheres that they put in with the islets, and therefore they can base the size of the islets that they're looking at on those microspheres that they know. There's also some, this is just something it was from Northwestern University a couple years back. It's an image analysis where they, the computer sizes the islets, and then but you still have a little bit of a personal thing of you have to go in and make sure they're counting actual islets and not just other tissue. There is some the same kind of idea of drawing the islets, getting the elliptical shapes, and this is something that came out also about 5 years ago that showed the difference between the accepted practice of counting islet equivalents and the difference with a spherical, more of an elliptical shape and not a total volume. So the purity estimation is something else that we do, and this is really pretty much an estimation. There's been automated ways of doing this as well with the image analysis that I showed in the last couple slides. But most people, especially if, like I said, if you're just doing research and you're not trying to transplant or you're not trying to distribute huge, large numbers of human islets, you're just going to estimate your purity. So there's high purity and there's low purity, and you can see the difference between these. Again, remembering that the islets are the little red dots. Purity also sometimes isn't just an isolated islet, but you'll see islets that have acinar tissue still attached to them. So that also has to be considered in your estimation of purity, depending on what science you're doing and depending on whether it's important or not. So everything really does come down to your own particular studies, but these are just some background information. So again, here's a estimation of purity. These are photos of islets that, and you can see in here, like right here, the whites, little whites, are acinar or other cells, and of course the red are just the purity. And I don't know what you would estimate this purity at, but and this was based on a number of different, the study that was done with a number of different investigators that had a lot of experience in counting human islets. I would say it was about 90%. And the reason you look at it, even though it looks like there's a lot of little stuff there that isn't islet material, that isn't red and stained, what you want to do is you want to take the total volume of the islet material versus the total volume of the other tissue, and that's how you would determine a purity. Again, with this one, this looks like there's some really big islets there, but we would probably estimate this one at about 70%. And this one's kind of a tough one. And this is a picture. These aren't stained islets. This is a photo of islets that were shipped actually by one of the distribution centers to an investigator, and showing the investigator said these are totally contaminated islets. They're horrible. This really is a pretty lousy prep, but it probably didn't start out with a really bad contamination or impure prep. What happened is the center had said it was like a 90% pure prep, and when it arrived at the center, they were saying, look at all this stuff that's in here. And what actually happened is that this isn't acinar tissue, this isn't acinar tissue, this isn't acinar tissue here, even though it looks, it's not islet tissue. What's happened is that the cells have broken down, either if it's correct for the center that shipped it, that it would be 10% of the tissue that was in there, which was acinar tissue, is broken down, or maybe it's islet tissue that's broken down. So these are all single cells that have broken down and caused what looks like a very impure prep. And these are those cells, or different cells, that have clumped together and has caused what looks like an impure prep. So that's something, if you're receiving islets from centers and there's a discrepancy between the purification, you need to make sure, take pictures, verify everything, and we'll help you out to try to, sometimes we can try to determine by photographs what's really going on. So anyhow, this could have been a 90 or a 95% pure prep, but because of the breakdown of cells, it doesn't look like anything somebody would want to use for their research. The next thing is viability. In our studies, we use, in our centers, we recommend to use the fluorescein diacetate and propydium iodine. Fluorescein diacetate is a stain of green, and propydium iodine stains red. One thing to be careful of, there will be a couple more of these here. The fluorescein diacetate is an inclusion dye, and propydium iodine is an exclusion dye. So again, viable cells are green, dead cells are red. Normally we would count between 50 and 100 islets in the in different categories, and then those would determine the entire viability of a prep. You probably won't do this, but one thing, if you do use this for dye, if you do use dyeing for your own studies, be careful that the fluorescein diacetate does tend to overpower the propydium iodine. So if you have, if you're only looking at one filter and you're just looking at the combination of the two, that is a little difficult. Yeah, Horatio?
So why FDA versus AOPI? The PhD thesis that looking into this was done by Nick by London like 20-something years ago, and the original was AOPI. Did you see any advantage of this?
I can just tell you that the reason we ask, and we don't require our centers to use these, but these two dyes, but it was something that was required for the CIT studies, and since a lot of our centers were part of the CIT.
But why was determined, or was it just an arbitrary thing? Because there is no advantage. I compare it side by side, and there is no advantage of one versus the other.
And and so there are a few other stains. There's the acridine orange, and there's another one, the
Green,
yes. So
there are many.
There are many, and all I'm saying is that this is how you will get your results from our centers, because, and the reason why we went to the CIT.
Yeah, but the CIT make a lot of arbitrary decisions. I wonder if they're...
I have no idea what, because we weren't part of the CIT, so I don't know why that was finally decided upon.
Okay.
Yes?
We switched actually from the FDA to just using Disazone. Because if you use Disazone and a PI, you can actually see the islets themselves, which are, you can see the rubidium iodide of each islet. You can distinguish it between the aster tissue, and you're getting an actual viability of the islets themselves.
Mhm.
And if you put FDA with PI, they fight for.
But this is not a vital stem. You're
Right, the PI and true, but it shows you which are islets, so the PI will show you exactly which islet is viable.
And that's very true, especially if you don't have a real pure prep. I think that's probably something that's really important. And if you're not that familiar with it, because it is sometimes hard to distinguish the difference between an islet and aster when you're doing the fluorescein stains.
Well, it's important to be able to document it too.
Sure.
So you can take those pictures and you can see which is the islet and
Mhm.
drawing representative of that particular islet.
If somebody is interested to create standards, I publish in analytical biochemistry, and each coded with different amount of zinc to standardize the premise. And the methods and everything are published in two papers, the zinc sensor and zinc content in bits in analytical biochemistry.
Okay.
10 years ago.
All right, thanks. Does anybody else have anything they want to add? Yes?
So basically, I think for the autologous islet, it's more critical use DTZ plus PI because we want to judge the viability of the islet instead of the whole mixture of a lot of other cell types.
That's very true. And I think most people are interested in just the viability of the islets. And you're right, if you have a prep that isn't very pure, then your viability estimates need to be adjusted, I guess, depending on what you're using. If you're not using, and I would say that most people use these both, but using them together, like you said, for documentation is a nice idea. Do you use, when you do use your Disazone though, do you use it, how do you mix it? Do you mix it with alcohol or do you mix it with DMSO when you make your Disazone stain? Do you know? DMSO? Because that in itself, couldn't that affect the viability of what you're counting?
Well, I think if you take too much time to do it, absolutely.
Yeah.
But you're turning it over as soon as possible, so
Okay.
I don't really worry too much.
Because I do know that in the past people had used ethanol to mix it, when they wanted to use those specific islets, not a sample, but those specific islets, because then it would, the metabolism would allow that to leave where I don't think that the DMSO, there's damage when that happens, so.
Well, it's also the islets are degranulated. They're going to look different because the EPC binds to the zinc. So the islets are degranulated because they have no insulin, like you see many times.
Right.
You look different.
Yes, but that also will give you an idea of that quality of that islet then too, I think. If you've got a type 2 possible.
Not necessarily. You can have great islets that are degranulated because of this circumstances, and they still have good islets, and they can reverse the other in vivo.
Yes, after a while, you're right. Okay, anything else? Okay, so once again, here's some estimations of viability of islets, and you can see that sometimes it's a little bit difficult to see the red here with fluorescein. These are just some nice pictures of islets and other tissue that has been stained. Now I'm going to go really quick through this human islet culture and what we use in our program for shipping. We use CMRL, basic CMRL with heparin, serum, either AV serum or fetal bovine serum. Some centers, it depends on which center you're getting those islets from, and you can have the choice of getting one, you can specify if you don't want to bring animal products into your facility, or if your experiments are going to be affected by human serum or by animal serum, you can choose to go with it one center or another. Right now we're using one shipping bag, which is an origin culture bag, and we put 120 mils, no more than 20,000 islets in that. We are looking into getting smaller bags that would hold less islets, so there isn't a loss, because there are some people that only want 500 or 1,000 islets, so we don't want them to get lost in that 120 mils of media. And we ship at room temperature. We did a number of shipping studies. Dr. Joyce Nyland is going to present a talk this afternoon at 4:30, I think, 4:45. Okay, and she'll give you some background on how we came up with this protocol for shipping, but it was done with a number of centers and a number of investigators a few years ago, and the paper just actually finally got printed last month, I think. There will be islet loss during shipping. If you get islets from us, there could be islet loss. We hope not. Just like you'll have islet loss during culture, and that can be based on the cold ischemia time of the donor, or oxygen deprivation during tissue harvest, how the pancreas was preserved, the islet index, then also culture temperature makes a difference. We require that the islets are held for 12 to 18 hours, actually, and we have a few caveats in that, but we want to make sure that when islets are shipped from the center, that they have already gone through whatever loss that they would possibly get due to the isolation processes, and have kind of settled down and reestablish any kind of dispersion that has happened during the technique of isolation and purification, and clumped, solidified a little bit better. Also, anything that was damaged dies off, and then when they're shipped, you won't see that damage. That's why we wait the 12 to 18 hours. You can culture islets up to 10 days. You can culture islets for up to 4 weeks. There's studies that were presented that go out even longer than that. It doesn't mean your islets are going to be the same as they were when you first isolated them, but you're going to get some central necrosis depending on the size of the islets over time. But you still will have some viable cells in your islets after a long time if you don't keep them at 37 for a very long time. As far as assays, this is just some potency assays that are used by different centers. We require glucose-stimulated insulin release just to show our investigators that there is some kind of testing that has been done to show that there is some potency in the islets that we're shipping. You may not get that information before you receive the islets, because most centers wait a day or two before they do their glucose stimulation assay, but you will get that information at some point. And if that's to be able to compare your studies with how the islets were before they were shipped from the center. You can do a mouse bioassay, which is a transplant, which is, it really correlates pretty well with transplant success in humans in clinical studies, but it's very expensive, it's a lot of work, and you have to wait for your results. And you have to have mice ready to go. Yes?
Do you have any studies from all the shipping that has been done, in which they culture the islets and perfuse them fresh versus 24 hours, see the differences?
Do I have any?
Maybe not you, but you are collecting on the data.
Yeah, I can't point that out, but I just know that from experience, It really depends on your isolation and your donor, but I think that in most cases, that rest and that 37-degree culture for at least a few hours after the isolation does help to make better results.
Yeah, but you don't have any data.
I can't think of a paper specifically right now. Maybe somebody else can. How about John? Can you think of?
There was an Argentinian boxer that said that the experience is like a call that comes to you when you are bored and you have the experience. You don't have any
Okay, well, thanks. Jonathan, can you think of anything that's about how to, if islets that are perfused immediately or assayed immediately after isolation have less results than if you keep going on culture?
We always used to keep our islets at 37 degrees for a minimum of 18 hours before perfusion, GSIR.
I know, but what hurts
That
just,
was allowed to allow the islets to recover from the insult by the
Okay.
isolation.
But there is no data actually, you can actually
No.
perfuse those cells, incubate the same set of cells, and 24 hours later, assess the same cells, the same or something like that. But from all the sheep islets and all the studies, that data doesn't exist.
no, I'm not going to say that data doesn't exist. I'm going to say that I don't know I don't know the name of the paper or the author of somebody that has proven that you have to hold the islets for 18 hours.
That's not my problem.
Yes?
Actually, we shipped a lot of items from the University of Virginia, and it was always most successful. I really got it if we kept them overnight. You know, there were some people that said we have to have them immediately, and there was a lot more islet loss than if we left them overnight.
So, there's anything else on that? I mean, you can talk about this if you want. I mean, obviously there's a little controversy, and maybe you could give us a paper that shows that it does make a difference to ship them early, or it doesn't make a difference. Another assay is oxygen consumption rate, and this is something that's relatively newer than some of the other assays, I guess. And a lot of people think this has got great potential for showing transplant success for islets. And of course, that would be the same then for your studies if you need to do it. But it's a little touchy of an experiment, and unless you're doing a lot of this, and that's what you're trying to prove, or you're trying to do clinical transplants, I'm not sure that it would be something that would be appropriate for people that are just using islets for a specific research project. There's microfluidic assays, which is kind of like a conglomeration of all these together with some other assays that are done by some universities. There's laser scanning that can actually pick out and measure a beta cell potential, and there's HPLC that are done by other places. This is just some examples, and I don't think that there's definitely, I'm not going to make a suggestion as to which one is the best, because that's really up for controversy. But different centers use different assays, and they're happy with those results. So what we do at the IADP is that we require a GSIR, a glucose-stimulated insulin release assay, to go with any islets that are being distributed through our program. And this is done, we've adopted the assay that was required by the CIT studies, the clinical islet transplant studies that were conducted by NIH, only because, like I said, it was something that was already documented. It had gone through a lot of vetting, and we said, well, if it's good enough for them, we'll go ahead and ask that to be our requirement. It measures the insulin output either by ELISA or RIA, and of course the stimulated insulin, the stimulation index. I don't know what this means, but it's not mine. So the insulin stimulation index is the measure of the high glucose divided by the lower glucose insulin results. Do I need to? Yep.
How typically are you doing, Julie Carnanti and Neil?
I'm sorry, I couldn't hear you. Hi, Julie.
Hi, Julie Carnanti and Neil France. I was wondering how physically you're doing GSIS. We normally are doing it 6 replicants and a 96 well plate with a filter on the bottom.
Mhm.
Sell them anymore. So are you doing it in an insert or like an Epidur?
They actually, yeah, the exact protocol is on our website, so you can see that. Anybody can see that, and we can give you that, or you can see it from our brochure. But it is in well plates, and it was what was adapted by the approved by the CIT studies. So it is in the well plates. It's not in tubes. It's not, and it's where you take the filters out, the little filters out, and move them from well to well. And like I said, you can find that on our website. A stimulation index for human islets is anywhere between 3 and 6. It can be a lot lower, it can be a lot higher, but I think that's something that's accepted. This is our website. This is our homepage of our website. We have a lot of information on there. If you become an investigator, this is the information that you would receive. You would get an email that we're doing a broadcast, or that one of the centers is doing a broadcast, and we do a matching to your requirements that you've already listed as an investigator, to an algorithm that that decides who's up for islets at that time, from which center. Dr. Nyland, like I said, will be giving a talk a little bit later on today that's going to give a lot more details. But you can click on our website. You can learn more about the offer itself, which is where it has all the donor shipping culture information for that prep. There's the initial broadcast data, which says the purity, the viability, and then there's final QA assessments, which assures that there wasn't any contamination at any point either, and that's required at 10 days after isolation and at 30 days, because like fungal contamination takes a lot longer to show up. Of course, if anything comes up before that, we let you know right away. This is kind of a list of the data, and it's really small. I'm sorry you can't get it much bigger, but you're welcome to look at the website and see this data. We give you information about type 2 diabetics if we have them, shipping information, and also one thing that we've started doing just in the last year or so is that if you are an investigator that gets islets, and we give you a list of all the other investigators that received islets from that same shipment. And you can go to our website and see what they're interested in, what their studies are about, and it gives you an opportunity to maybe make collaborations with another investigator for studies that could match up. You can also do this ahead of time. If there's, you can look, and if you know someone that you want to do a collaborative study with, we will put, when your number comes up for broadcast, when you're picked by the algorithm to receive islets that are broadcasted, we will automatically match your collaborator to get those same islets, who may be doing a different study totally, but you're trying to write a paper where you can use the science from both centers to come up with a scientific end. So you can apply for that beforehand, or if it's something that comes up to you a little bit later, you can see every time you receive islets who else has gotten those same islets. If they're a buddy, you can call and say, hey, how did they work for you? Because they didn't work for me. Or hopefully it's more like they worked wonderfully for me, and my paper is going to be published now because everything was so successful. And they'll say, yeah, same for me too, and here's some more data. So anyhow, this is just one of the things that we try to help out with. We also, the quality assessment is always given to each investigator after they receive the islets, which also has the glucose stimulation index, and as well as all the QA data, all the bacterial or fungal contamination data. That's it. I don't even know what time it is. I don't have a timer thing up here. Pardon? On time?
29.
Oh, okay. Wow, that was good. If anybody has any questions, you can talk to me now, but since we only have 1 minute, I guess you can't say much. If you, on your way out, well, first let me introduce a few people here that's in part of our study. Dr. Joyce Nyland is right back there. She is the program director of the IIDP, and she will be giving a talk again at 4:25. Is that what it is? I can't hear. 4:45. Yeah, right before the welcome committee. So, you know, you'll be really thirsty after that, and you can go get a glass of wine. And then Martha Antler is standing in the back. She takes care of so much stuff, I can't even go into that, for the IIDP and another consortium that we work with at City of Hope. Cindy, I almost said Cindy Palmer, Cindy Crosby, it takes care of all of our financial management, and if you become part of the program and subscribe to the islet program for human islets, you'll be talking to Cindy, I think, as well as James Cravens. James is right there. He's kind of the investigator problem person. And then Janice Sawinski is right there, and she's our I don't know all the titles. She's our program manager? Administrator. There you go. And I'm Barbara Olack, and I work mostly with the centers, but if you have any questions about islets, I've been doing this for a long time, and I've seen a lot of problems and a lot of good things over the years. So maybe I can help you out. Feel free to call anytime. And my phone number and email address are on the website as well as everybody else's. So please, if you want a training book for your lab, they'll be up there, or are you giving them out? They'll be up there at the back. Please feel free to pick one up if you need it. If we run out, just give us your name and address, and we'll ship you one. And also make sure that you sign the sign-up sheet so we know who came, how successful this was, and if we should do it again. This is like the 10th one, I think, that we've given. So it just keeps changing a little bit every time. So if you've been here before, sorry for the repeat.
And Barb, soon you will have a version of this posted on our website.
Yes.
Voiceover, PowerPoint.
That's true. We actually have the slideshow on our website, but the new one will be up on our website. There's older ones on the website. And I'm going to do I'm not exactly sure if it'll be a podcast or it'll be just a talk through of the slides, so that if you have new people in your lab that you'd like to refer this to, you're welcome to. Plus, I think I was told that the IPITA people are going to have this for the delegates on their website as well, a telecast, like they're recording back there right now. So everybody can wave as you walk out, and you'll be on there. All right? Thanks a whole lot for coming.